Hello, my name is Chwee. I'm currently a professor and director of the Institute for Health Innovation and Technology at the National University of Singapore. I'm also an inventor and entrepreneur, having co-founded 6 startups to commercialize technologies developed in my lab. When the Berlin Wall fell, I was a final year undergraduate student studying mechanical engineering at NUS here in Singapore. I followed the news with great interest as I was planning for a 5-week backpack trip to Europe and was wondering to myself if I could finally visit East Germany. As a child, I was very fascinated by technology. For example, how does one design and build a car or an airplane? In fact, during that time, there was a number of movies that really motivated me to become an engineer. These include Back to the Future, RoboCop, and James Bond's movies that featured some really cool futuristic technologies, which I thought will one day become a reality. Looking back, I'm glad that I've made that right choice. I'm thankful that I can now have the opportunity to use my engineering skills to develop medical technologies that can directly benefit the patients and the society. We are in the midst of an unprecedented COVID-19 pandemic that is not only disrupting lives but also livelihoods. As such, rapid on-site testing is key not only for us to contain the disease but also to identify individuals that may be infected so that we can quickly isolate and treat them. Unfortunately, current tests requires the sample to be first collected, then sent to a centralized lab for testing, and it can take up to a few days before the result is made known. The current gold standard test for COVID-19 is the polymerase chain reaction or PCR test. This test involves taking the viral RNA and then replicate them to an amount that is detectable. In our lab, we have developed Epidex, a portable, low-cost, point-of-care micro-PCR system that can enable us to perform tests on COVID-19 samples from 1 hour to less than 30 minutes. What we hope to do is to break the wall of slow response to disease outbreak by bringing this rapid diagnostic test to the masses for on-site testing with immediate delivery of results. Shortening the time of diagnosis requires us to be able to re-engineer as well as reinvent current testing methodologies. As compared to the conventional PCR test, our Epidex micro PCR system has several innovative features. Firstly, it makes use of a microfluidic chip to perform both the RNA extraction and PCR process all on one chip. This reduces the time for the test. Secondly, the amount of reagent used is half of that needed for the conventional PCR test. As such, we are able to reduce the cost of the test as well as mitigate the problem of reagent shortages. Thirdly, the microfluidic chip has a small footprint, which means we can build a smaller and more portable PCR system that can be quickly deployed on-site for immediate testing, whether in a school, workplace, or the airport. We have seen how this slow response to disease containment can adversely impact all of us in this closely interconnected world. One of the reasons for this slow response is the lack of a rapid on-site diagnostic kit to quickly identify and immediately isolate and treat infected individuals. We hope to break the wall of this massive outbreak by making available an affordable, portable, and rapid on-site diagnostic test kit. I envisage our micro-PCR system Epidex to play this important role, not only in curbing the current but also future outbreaks. There are still many unanswered questions with regards to COVID-19. For example, why do some people get very sick and some don't when they are infected? Are asymptomatic patients infectious? Will COVID-19 become endemic? When are we going to get a safe and effective vaccine? As such, much research still needs to be done before these questions can be addressed. Nevertheless, our response to COVID-19 has to be based on these scientific evidences and outcomes.